So you guys, if we're talking about mycobacterium, like having the most resistant cell wall, on the opposite extreme are a group of bacteria which lack cell walls. They don't have cell walls. And the classic example are the bacterial pathogens called mycoplasma. Now watch out, you guys, because if you're speed reading, might you see myco and think mycobacterium, and vice versa. Over here, you're speed reading, you see myco, you think mycoplasma. Force yourself, you guys, to read the whole word, right? Because mycoplasma lack a cell wall. Mycobacterium have like the most resistant cell wall of all the bacteria. So these mycoplasma, you guys, since they don't have cell walls, they don't have peptidoglycan, they're in danger of osmotic lysis. So what they do when they're living in us, for example, they can cause pneumonia, walking pneumonia. They steal cholesterol from our cells and insert it into their cell membranes to strengthen their cell membranes to prevent osmotic lysis, right? So the mycoplasma, they steal cholesterol from the human host. And where do they put it? In their cell membranes, right, to strengthen the cell membranes so they won't undergo osmotic lysis. But you guys, it's a little bit tricky because this statement is true. Bacteria cannot make cholesterol. That's a true statement. But is there a group of bacteria that you would find have cholesterol in their cell membranes? And who would that be? The mycoplasma. How they did it? They stole it, right? They stole it. Okay, so you guys, we're going to blast into Unit 3 here. So you guys, this is on, the unit is on cells, but because most of you have had or will have 2 semesters of anatomy and physiology where you're just focused on eukaryotic cells, human cells, I'm going to focus primarily on bacteria cells, because I don't think you're going to get this information probably in most other classes. And one we'll be stressing is antibacterial drugs, antibiotics. What are their targets? How are they going to kill the bacteria? Okay. So in general, you guys, when we're talking about cells, there's some characteristics of cells that all cells share, and this is just a few of them. So all cells, you guys, synthesize the cytoplasmic membrane or cell membrane or plasma membrane. All cells have ribosomes. What's the function of ribosomes? Protein synthesis is good. All cells carry out metabolism, so one example would be all cells can make ATP. Cells grow and divide. And these, again, are just a few characteristics. But you guys, this is really important. Viruses and prions are not cells, and if I asked you for proof or evidence, you could say, well, viruses and prions can't synthesize the cytoplasmic membrane, the cell membrane. Viruses and prions lack ribosomes. Viruses and prions do not carry out metabolism. Viruses and prions don't grow and divide. So viruses and prions, they violate all these characteristics of cells. And folks, this is just reviewing our phylogenetic tree of life, a cartoon that represents our understanding of how life evolved. And you guys remember the unity of cellular life. As biologists, we describe that as a result of all cellular organisms being descended from the same, common ancestor, that last universal common ancestor, Luca. And that helps us understand why even bacteria and human cells, they share many similarities, right? But then the diversity of cellular organism is described through biological evolution, right? And one of the driving forces being a natural selection, where over time, the variants in the population that are best adapted to their environment will survive, reproduce at higher rates, pass on those variant genes to their offspring. And that means, as a result, through biological evolution, populations of organisms become better adapted to survive in any given environment. And remember, guys, the environment's constantly changing, so we really need that genetic variation and natural selection to continue. So you guys, what are these 3 big groups of cellular organisms? these are the domains, right? You guys, and so which two domains have prokaryotes? Domain bacteria, you guys. Which domain has all the eukaryotes? Eukaryote. And you guys, what are these lines? These horizontal lines that start that originate in domain bacteria, and then they end up over here in domain eukaryote. What the heck? What's going on there?
That's chloroplasts.
Okay, you guys nailed it. Yeah, this is kind of a cartoon of the endosymbiotic theory, which states that eukaryotic mitochondria evolved from primitive aerobic respiring bacteria, and eukaryotic chloroplasts evolved from what? Primitive cyanobacteria, right? Isn't that cool, right? So you guys, all of our cells that have mitochondria in them, all those mitochondria are descended from bacteria. Yeah, that's just, I know, I'm easily amused. I just think that's the coolest thing. Okay. So folks, again, we're going to be focused on bacteria cells. So we just want to remember they're prokaryotic cells, which means they lack a membrane-bound nucleus, and they lack other membrane-bound organelles. So prokaryotes don't have mitochondria. Prokaryotes don't have chloroplasts. Okay. And you already told me it's domain archaea, domain bacteria, and we're going to focus on bacteria. Okay, guys, I know this was a horrible slide. I apologize. But I put it in there to remind me to tell you guys that this diagram is probably going to be on the lecture exam. And what I'll do is I'll white out all the labels, all the descriptions, and I'll use letter labels, and then I'll have you answer some questions providing the letter label of the correct structure. So it might be I'll describe a function and you tell me which structure is involved in that function. It might be I will describe the structure and composition and you'll tell me which structure matches that description, okay? And furthermore, folks, since you know the exam is coming up on Thursday, I want to first go over this generic bacterium and give you an overview of structures and functions, because this will provide a really strong foundation for the bacteria part of our course, of this lecture rather, right? So I want you to be able to think about this over the weekend, so you won't be cramming it, you know, like Wednesday night, okay? So you guys, what we're going to do, oh, and furthermore, you guys, sorry, I would really recommend, if you have colored pencils, to color the different structures, and especially, guys, these layers. See, these layers get really confusing, so if you use colored pencils to color them different colors, I think it'll be easier when you look at this cartoon on the lecture page. So folks, what we're going to do is we're going to start, and again, guys, this is kind of the big overview, right? We're looking at the course, and then we'll get down to the view. So guys, what we're going to do is we're going to start with the living boundary of the cell, which is this innermost kind of thin structure. This is the cytoplasmic membrane, or cell membrane, or plasma membrane. And the most important function is to control what moves into and out of the cell, right? Remember, you guys, fluid mosaic model. So we understand the structure is a phospholipid bilayer with proteins. Okay, all right. And now what we're first going to do, you guys, we're going to move to the outer layers and outer structures, and then we'll zoom in to the inside of the cell. So you guys, this next layer out here, a little bit thicker, this is the cell wall. And the most important function of the bacterial cell wall is to prevent a phenomenon called osmotic lysis. Osmotic lysis is when water flows into the cell and blows it up like a water balloon until finally the cell breaks open, or lysis. Right? So the diffusion of water is described as osmosis. So we call this phenomenon osmotic lysis, when the cell breaks open because of water moving in. Okay? So the job of the cell wall, you guys, is to prevent osmotic lysis. And we'll come back to you guys and spend quite a bit of time developing that idea. To prevent osmotic lysis, the cell wall has to be strong. So you guys, what is the component in bacterial cell walls that provides strength? Peptidoglycan, right. And you guys, we're going to go crazy. We're going to go over the structure of peptidoglycan, because we know that if we could prevent proper synthesis of peptidoglycan, say we're treating a patient with a serious bacterial infection, if we could somehow interfere with the ability of those bacterial pathogens to make peptidoglycan, we could kill them. And how could we do that? Through osmotic lysis, right? So we're going to spend a lot of time developing the concept concept of how antibiotics can interfere with glycan synthesis and then kill these disease-causing bacteria. Folks, this outermost layer, let me give you a broader term for it. The broader term for that outermost layer would be glycocalyx, and I'll write it on the board. It literally means a sugar cup, and it's because very often these outermost layers are made out of polysaccharides or sugars. So glycocalyx is a more general term, you guys. It means sugar cup, or like a sugar coat, and it's because it's usually made of polysaccharide. There are some weird exceptions, you guys. So it's usually made of sugars, polysaccharides, and there's two basic types. There's two types depending on how well organized they are, how tightly adherent they are. The first one, you guys, is called a capsule. And capsules are really important for bacteria that are going to invade humans. Because the bacteria, right off the bat, our phagocytic cells are going to try to attach to them and ingest them through phagocytosis and kill them. So the beauty of a capsule, if you're a bacterium, is our phagocytes can't attach to it. Our phagocytes don't have any receptors for these capsular polysaccharides. So the phagocytes are trying to latch on to the bacterium, but if it has a capsule, it just slides right out of the grasp of the phagocyte. So you guys, we say that capsules then are anti-phagocytic, or they inhibit phagocytosis. And again, that will be really important for bacteria that are invading, say, a human or another animal. Okay? The second type of glycocalyx, you guys, is called a slime layer. And slime layers are often sticky, and they permit attachment to surfaces in the bacterium's environment. And the great example, you guys, we're going to see next week is we're going to do tooth scrapes. We're going to scrape some of the bacteria living in a biofilm on your teeth, and the biofilm that's formed on your teeth is called plaque. We're going to scrape some of that, smear it on a slide, and gram stain it so you get to see your cells of the bacteria that are living with you. Prevent attachment to surfaces, and we'll see you guys, these slime layers are important in forming biofilms. And we'll come back and talk more about biofilms. But remember, you guys, your teeth plaque is a type of biofilm. So we'll just put that, for example, plaque on your teeth. Okay. All right. And again, you guys, it's very, very superficial, right? We're just kind of getting to the big view here. So next, you guys, we're going to look at these little thread-like, hair-like structures protruding from the surface of the cell. All of these filaments are made of protein. Okay? And this first one here is called a fimbria. So let me see what we can do this. Okay. So these little protein filaments, folks, sticking out of this cell. You could do three types. And the first one, the fimbria. And these are proteins that are used to, again, attach to surfaces in the cell and sometimes attach to other cells. And an example would be other cells. And the at the tip of the fimbria, you guys, are going to be special proteins called adhesins. What does adhesin sound like? Sticky. Sticky, like adhesive tape, right? Sticky. So that's kind of telling us that the adhesins are going to bind to specific molecules in the environment or specific molecules on a cell. And that's how they'll stick to them. And one example, you guys, that is, to me, is like, okay, that makes sense. Let's say that an E. coli that normally lives in our intestine, right? So our feces are going to be chock full of E. coli. Let's say accidentally you contaminate the opening of your urethra with some feces. Well, what E. coli can do, it's motile. It can ascend, move up your urethra to where? Your bladder, right? And like E. coli is like, this is a cool place to hang out, right? But what must the E. coli do to live in the bladder, to colonize the bladder? What's going to happen when you urinate? It's going to get washed out, right? So E. coli has these filaments with adhesins that permit it to attach to special molecules called receptors on the surface of our bladder epithelial cells. And that's how it sticks in the bladder so it won't get peed out, right? So a lot of times, you guys, pathogens colonizing other organisms, they have to find a way to stick to those host cells, otherwise they're going to get washed out. Okay? So that would be one function then of these little vibrio, to help them attach to surfaces or cells in the environment. This next one, you guys, is called, it has several names. It can be called a sex pilus or a conjugation pilus. And these play important functions in a process called horizontal or lateral gene transfer. Now, as humans, you guys, we're accustomed to the idea of like us transferring our genes to our children, right? So that would be an example of like vertical transfer from one generation to the next. But bacteria are wild, you guys. Bacteria love to share their genes with their neighbors. So Vanessa, here's an antibiotic resistance gene for you, right? So bacteria can share genetic information with their neighbors. That's kind of bizarre to us. I mean, the only way we could do it is like some genetic engineering, something artificial. But in one type of horizontal gene transfer, you guys, called conjugation. We use terminology a lot like blood donors or organ donors. One bacterium, this bacterium is going to be the donor bacterium. And then we're going to have a recipient bacterium. And this donor has some DNA, copy of its DNA, that it wants to transfer to the neighbor, the recipient bacterium. But to do that, you guys, they have to make physical contact, right? They have to touch one another. And that's the job of the sex pilus or conjugation pilus. So the donor forms this sex pilus or conjugation pilus to attach to specific receptors on the surface of the recipient bacterium. And it's wild, you guys. Then the donor bacterium can disassemble the sex pilus, and that draws the recipient in close physical contact. And it's then that the donor can transfer the DNA to the recipient. Okay, so that sex pilus is how the donor is going to make physical contact with the recipient and draw the recipient close. So the donor can literally inject the recipient with a copy of DNA. And the bad news is, you guys, it could be like antibiotic resistance. Okay. This last filament here, you guys, these represent, if we had a single one, you guys, it'd be called a flagellum. Or if we have more than one, so flagellum, you guys, is singular. If we have more than one, it's flagella. That's plural. And these are structures bacteria use for motility. What does motility mean? Yeah, the ability to move. Now, you guys, not all bacteria have flagella, but it's a huge survival advantage, you guys. Because let's say you're a bacterium living in a liquid environment and you need nutrients, right? So it's really helpful, you guys, if you can move towards a source of food, nutrients, right? That's going to increase your survival. And the reverse is true, you guys. What if there's some poisons or toxins in your environment, right? It's going to help you survive if you can move away from them, right? So motility is a wonderful survival trait. You guys, the flagella, they're really cool. They're anchored in the cell membrane and cell wall, and they rotate like a boat propeller. It's just like a boat propeller, and that's how they move the bacteria through their environment. And again, folks, we'll come back and add more detail in later slides. So you guys, we work from the cell membrane outward. Now we're going to go inside the cell, okay? And there's something there that looks like a piece of spaghetti. What do you think that spaghetti, tangled spaghetti is? Yeah, that's a chromosomal DNA. Okay. And what's wild, you guys, is in bacteria usually have a single circular chromosome. And to me that's kind of wild, because what kind of chromosomes do we have, you guys? Humans have linear, multiple chromosomes. So bacteria just do everything different. And the composition, you guys, would be double-stranded DNA. Okay? And the double-stranded DNA, you guys, it carries all the genetic information for the cell. And one example, you guys, is we kind of talked a little bit about this. The DNA sequence provides the information for the amino acid sequence of the cell's proteins, right? And those proteins will be important in building the cell. And the proteins are also important because they're going to become the little biochemical tools of the cell, the organic catalyst that will make all the other components of the cell. Yeah. Okay. So that's the single circular chromosome. There are always exceptions. You guys, there are some bacteria that have like linear chromosomes, but we're talking about the average generic bacteria. And then folks, see this little circle right here? That little circle is what we call a plasmid. And so plasmids are made up of double-stranded DNA. They're circular. They can be self-replicating, so they don't have to wait for the cell or the chromosome to replicate. And they usually carry extra genetic information. And the extra genetic information we're really worried about is antibiotic resistance genes. So such plasmids, you guys, that carry antibiotic resistance genes are called resistance plasmids or R plasmids. And what's really frightening, folks, is that some of these resistant plasmids, they can carry 4, 5, 6 genes for resistance to 4, 5, or 6 different classes of antibiotics. And what makes this even more nightmarish is, you guys remember this horizontal gene transfer here? Very commonly, what gets transferred from the donor to the recipient is a copy of an antibiotic resistance gene. And if that gene is carrying information to resist 4, 5, or 6 different types of antibiotics, you know, boom, in just that little tiny event, this bacterium then now is resistant to 4, 5, or 6 different antibiotics. And this is a huge problem, you guys, in environments where a lot of antibiotics are used, and probably a lot of you work in those environments, hospitals, right? Yeah. So huge concern for us humans, this lateral transfer of these antibiotic resistance genes. Okay. What else is here? Oh, these little dots, you guys. Those little dots. Those are ribosomes. You guys, you told me before. What is the function of ribosomes? Yeah, awesome, you guys. They're the sites of protein synthesis. And ribosomes, you guys, are made up of ribosomal RNA, or rRNA, plus proteins. And to me, again, you guys, I'm easily amused. It's so cool because it turns out the ribosomal RNA is the organic catalyst. It's the one that catalyzes formation of peptide bonds in the protein. I just know why. I thought, that's so cool. So the ribosomal RNA catalyzes formation of peptide bonds. And you guys, we have a fancy name for catalytic RNA that's acting as an organic catalyst. We call them ribozymes. So instead of enzyme, which suggests a protein catalysts, these are called ribozymes. So in the ribosome, you guys, it's ribozymes, RNA, that's catalyzing formation of those peptide bonds. Now, really, really important, guys, in bacteria have little ribosomes. And we measure ribosomes, you guys, by using Svedberg units, which are units used in sedimentation experiments. So just right now, you guys, without going into a lot of detail, the bacterial or prokaryotic ribosomes are called 70S ribosomes. And the reason we call those little ribosomes is that eukaryotes, like ourselves, we have big ribosomes in our cytoplasm. We have 80S ribosomes, they're larger. Now, I have to be careful in the cytoplasm. Now you're like, well, who cares? Well, you guys, this 70S ribosome then becomes a unique target for what? For antibiotics. And we're going to see a lot of antibiotics, you guys, that are targeting that little 70S ribosome of the bacteria. And it's cool because they can shut down the 70S ribosome without shutting down our 80S ribosome, right? We don't want our 80S ribosomes inhibited from making proteins. That would be really bad for us. Okay, so we'll come back and talk about antibiotics that will target the 70S ribosome. And then, folks, this last structure, thank heavens, not many bacteria can make this last structure. It's called an endospore. Before we discovered the disease-causing prions, endospores were used as the example of like one of the most resistant biological structures known. Endospores, you guys, is the reason autoclaves were developed because just simple boiling won't destroy an endospore. So let's just take a look at these endospores. And again, folks, we're so lucky. Most bacteria don't make endospores. And the two groups of bacteria, you guys, that contain important pathogens that we'll be talking about in the course is the genus Bacillus. And you guys in lab, which pathogenic Bacillus did we look at today? Anthracis, right? Right, and the whole genus Bacillus can make endospores. And you guys, the whole genus Clostridium can make endospores. So we already talked about C. diff, Clostridium difficile, Clostridium tetani. What does Clostridium tetani cause? Tetanus, right? Clostridium botulinum. What does Clostridium botulinum cause? Botulism, right? So there's several different clostridial pathogens, and sadly, they all make endospores. So what's the function of the endospore? Its function is to protect a copy of chromosomal DNA when environmental conditions deteriorate. Okay, so we'll make a story up, you guys. So let's say maybe this is like a Bacillus anthracis, and let's say maybe it's living in a cow, right? So anthrax can affect cattle, and let's say that the cows died, right? Decomposing. So this bacterium is probably going to die, right? The environment's getting bad. Maybe the tissue is drying up, right? So what will happen is that the bacteria will trigger a process called sporulation, forming a spore. And in sporulation, what they do is they quickly make a copy of their chromosomal DNA, and then they start laying down layers and layers of protein and peptidoglycan around that copy of the chromosomal DNA. And then furthermore, you guys, in the endospore, so we're going to have lots of proteins, peptidoglycan, and doing chemical analysis, they've tried to figure out why are endospores so heat resistant? Why is it we can boil them and it doesn't destroy them? And I don't know the biophysics behind this, you guys, but they've discovered that there's a high calcium ion content, lots of calcium ions, low liquid water content, and there's a unique substance called dipicolinic acid. And it's thought, and again, you guys, I don't know the biophysics behind it. It's thought this combination of high calcium, low water, and dipicolinic acid, this is what makes these structures very resistant. So they are resistant to heat, like we said. They're resistant to a lot of antiseptics and disinfectants. You guys are totally resistant to antibiotics. They lack our antibiotics, right? And as we said, they can remain infectious in the environment for years, decades, centuries, maybe thousands of years, right? So like we said, thank heavens, not many bacteria make these things. Now, in this example, you guys, where we had Bacillus anthracis forming an endospore, that endospore can remain infectious in the dirt for years and years and years. So let's say maybe maybe homes or shops are going to be built on what used to be farmland, so they go in and start digging up the dirt, and then that causes the endospores to be aerosolized, and we can inhale them and end up with ventilation anthrax. We could get them in cuts and have cutaneous anthrax, right? In parts of the world, you guys, where it's really hard for people to find good protein, sometimes they'll maybe find an animal that's died of anthrax, and they'll harvest the tissues, right? And of course, protein doesn't kill the endospore, so they end up with gastrointestinal anthrax, which is really bad, really bad. Yeah. So you guys, so remember the endospores, they're one of the tougher biological structures known, but it's important, you guys, will normal autoclaving destroy them? Yes, right. Remember, that was why autoclaves were developed, was to kill the endospores. So what they do is they use moist heat under pressure, right? And if you increase your pressure to 15 pounds per square inch above atmospheric pressure, right, the temperature is increased to 121 degrees Celsius. We usually autoclave for 15 to 20 minutes. That will destroy the endospores. Will that destroy disease-causing prions? No, right? What a nightmare for hospitals. Yeah.
Are the autoclaving conditions something that we should remember?
Yeah, you should. You should. That's something. As a microbiologist, that's a good value to remember. Okay? So you guys, just remember, 15 pounds per square inch of pressure above atmospheric pressure. So atmospheric pressure is already around 15 pounds per square inch. So you're just increasing the pressure like 1 atmosphere, and it's usually 15 to 20 minutes. This will destroy endospores, but it will not inactivate disease-causing prions. So folks, this next slide is just like, as a teacher I learned, the more sources of information you have, the better. And so I just put in a different artist's depiction of a generic bacterium. Just again, you guys, the more different sources of information you have, the better your understanding. You understand it more holistically. So that's just why I do this diagram in there, right? So now, you guys, this might be a little bit boring because we're just going to repeat what we said and just add a little bit more detail. But like I said, you guys, hopefully that's going to give you a strong foundation so that you go into the lecture feeling pretty confident about basic bacterial structures. Now we're just going to add some details. So you guys, so we're going to start again at the living boundary of the cell membrane, and the plasma membrane. What's the most important function, you guys? Okay, now that's going to be the cell wall, so that was good. So the cell membrane, and you know what, thank you so much, because I see that on the first exam. People get cell membrane and cell wall confused. You guys, maybe just when you're studying, do a simple cartoon to remind yourselves. Let's say, you guys, this is our bacterium. This is the living boundary of the cell. What is this? That's a cell membrane, cytoplasmic membrane. So you guys, all cells have to have a cell membrane. But most bacteria have something outside the cell membrane to protect it, and what is that? The cell wall. So you guys, just maybe cartoon it for yourself. Because on the first exam, I do see a lot of times people are mixing up cell membrane and cell wall. Okay? And remember, you guys, all cells have to have a cell membrane, but you can be a cell and not have a cell wall, right? It's not a requirement in life. What about your human cells? Do you have cell membranes? Do you have cell walls? No, right? That's one of the characteristics of kingdom Animalia. Animal cells don't have cell walls. But are we still cells?
Yes.
Yeah, okay. So thank you so much, because that is really confusing initially. Okay. So, okay, so what is the important function of the cell membrane? It's going to control what moves into and out of the cell. That's the most important function. There's other functions we'll discover later. You guys, what is a cell membrane made of? Phospholipid bilayer with proteins, right? And what's the model? the fluid mosaic model gives you guys. And it's really important, again, that those phospholipids remain fluid. If we damage the cell membrane, you guys, will the cell be okay? No, right? Because if the cell can't control what's moving in and out, usually the cell's going to die. And you guys, I do not ask this on the first lecture exam. It was just me being nerdy. And I know a lot of you are really interested in antibiotics. So 2 antibiotics that will directly damage or cause malfunction of the cell membrane, polymyxin B and daptomycin. This is sometimes used topically, you guys. A lot of us carry Staphylococcus aureus in our nose. So a lot of times they'll use topical daptomycin, so you would spread it at the entry of the nostrils to help try to destroy the Staphylococcus aureus that you're carrying. And of course, you guys, that would be really important. Let's say you're maybe a nurse on a burn ward. You don't want to be carrying Staph aureus and maybe infecting the patients, right? So sometimes they use daptomycin. And you guys, I apologize for the lousy, but it reminds me to tell you to turn to page 2. And it also reminds you guys, you might see cartoons similar to that on the lecture exam. So I never want you to be surprised when I put things on a lecture exam, okay? So, you guys, let me quiz you. These are cartoons of what? The cell membrane, right? Awesome, you guys. And then, what are these little guys that, I don't know, almost look like a little sperm right here? What's that? That's a phospholipid. What's that round part? The head, is it hydrophilic or hydrophobic? Hydrophilic, good. What are these squiggly little guys right there? Those are the hydrocarbon tails. Are they hydrophilic or hydrophobic? Good. So you guys, you could label here that this is the hydrophobic area right here. And what area is this up here? Hydrophilic and hydrophilic. Good. One reason, you guys, I like this little one is what are these squiggles? Those are membrane proteins. And the reason I like it, you guys, is that they're telling me, or indirectly they're telling me, that in our membrane proteins, amino acids with nonpolar hydrophobic R groups are usually going to be clustered in here in the hydrophobic region. And what do you think the R groups are like on the parts of the protein that are exposed to water on either side? What would your guess be, the nature of those R groups? They're going to be hydrophilic, right? They're either going to be polar or maybe ionized, right? Good. And you guys, if I showed this to you, and maybe I put like an A on the little circles, and a B on the squiggly lines, and a C on this guy right here, and then I asked you, which label structure would permit large hydrophilic molecules to cross the membrane? What would you tell me? C, right? Okay. If I asked you which structure, when hydrolyzed, would release amino acids, which structure would you choose? Okay, so remember, which structure, when hydrolyzed, would release amino acids? Protein, right? So it would be the same one, right? Does that make sense? If we hydrolyze a phospholipid, you guys, what would we get? We'd get fatty acids, a glycerol, a phosphate, and some other group, right? Does that make sense? Okay. And again, folks, just a little nicer concept map here showing phospholipids, the membrane proteins for transport, and just showing the location in a bacterial sketch, and then in this electron micrograph, here's the outer membrane, and then this portion out here, you guys, that's going to be the cell wall. Okay, folks, now this was cool because we already went over this a little bit. So remember, you guys, we said cell membranes, cytoplasmic membranes, they have to be fluid. And, the best description I heard was that cell membrane should have the consistency of like olive oil. And that's a problem, you guys, because that means the cell membrane is not going to be very strong, right? And this is a huge danger when our bacteria are living in what we call hypoosmotic environments. So let's develop this whole idea. It's hypoosmotic or hypotonic environments. So let's first develop this concept of a hypoosmotic or hypotonic environment. So what we're doing, folks, here is we're taking our little bacterial cell, and here, you guys, this is going to be the cell membrane. We're going to start with a bacterium. And all it has is a little cell membrane. Okay. Now, if I'm going to have it grow in a hypoosmotic, hypotonic environment, I need to know the concentration of water inside and outside the cell and the concentration of solutes inside and outside the cell. Okay, so you guys, if we use little dots to represent solutes, these could be nutrients, ions, waste molecules. So these little dots, these equal solutes. Usually bacteria are living in environments where there's not a lot of food around, so they're having to actively move food, nutrients into their cell. So generally speaking, you guys, that means that there's going to be more solutes inside the cell than outside the cell. So when we're talking about these environments, you guys, we're always comparing two compartments. So you guys, if we're comparing inside the bacterium to outside, which compartment has a higher solute concentration? Inside, right? So we're going to put high solutes, and we're going to put low solutes outside, right? Okay. What we're really interested in, you guys, is not, well, we are interested in solutes, but what we really want to know about is the concentration of water inside and outside. And you guys, solutes, they look little, but they take up space. So now what we want to do, you guys, is compare water concentration inside and outside the cell. So which compartment is going to have the higher water concentration? Outside, right? Yeah, good job, you guys. So we have low solutes, high water outside. Inside, high solutes, low water. Now, who cares? Because we need to think about osmosis. And osmosis, you guys, is the diffusion of water from an area of high water concentration to an area of low water concentration across a semi-permeable membrane. So you guys are little bacteria living in this hypoosmotic environment. What will be the net flow of water? Will water flow from inside the cell to out, or from outside the cell in? Out to in, okay. Now, this is a problem, you guys. This little bacterium is like a water balloon, right? And water is going to continue to flow into the cell. Water pressure will build up, exerting a force on that delicate little cell membrane. And what's going to happen eventually? Burst. Just like a water balloon, right? It's going to break open. What's the fancy term for a cell breaking open? Lysis, right? What caused it? Osmosis, right? So we call this osmotic lysis. Would it kill the cell? Yes. You betcha it's going to kill the cell. So you guys, because bacteria, they so often have evolved in hypoosmotic environments, there's been natural selection for a structure that will prevent osmotic lysis. What's the structure? Cells. You nailed it, you guys. So that's what most bacteria make, is a strong protective cell wall. So here, you guys, I'll use red for the cell wall. They make this strong cell wall outside the cell membrane. And the beauty of this is, you guys, is water is filling in and creating an outward force. The strong cell wall is exerting an equal and opposite force. And that's what's going to prevent osmotic lysis. So the most important job, you guys, of the cell wall, it's going to do what? Prevent osmotic lysis. Must the wall be strong to prevent osmotic lysis? You better believe it. It has to be really strong. What will provide the strength in bacterial cell walls to prevent osmotic lysis? Peptidoglycan. You've got it, you guys. Okay, excellent. The peptidoglycan provides the strength. Okay, so you guys, we're going to really develop this. Because remember how we said we're looking for unique targets for antibiotics or antibacterial drugs, right? And boy, is that peptidoglycan a beautiful target. Okay, so we're really going to develop this. Okay, so we know what osmotic lysis is, right? This is classically the experiment we use, these YouTubes. But to me, it's like, I like using this example better. This is more like real life, right? Like what we're interested in. So here you guys, here's a little cell living in growing and living in a hypoosmotic environment, undergoing osmotic lysis. And then this is the solution. You guys want the solution is to build that strong cell wall on the outside to prevent osmotic lysis. Okay, so I apologize. A lot of these slides are just repeating what we said already. Okay, so to prevent osmotic lysis, the bacterium will synthesize a strong cell wall outside the cell. And so you guys, we've already answered this. We'll explore in greater detail what the cell walls are made of, but the key ingredient is peptidoglycan. Peptidoglycan is what will provide the strength. So we're going to focus a lot on peptidoglycan. So you guys, we really need to understand the structure of peptidoglycan because we want to understand how to interfere with it to kill our bacteria. So you guys, on page 3, there's some diagrams regarding kind of basic subunits of peptidoglycan. So page 3 and page 4 you guys will be looking at, right? So you guys remember we had introduced the concept of modified glucose in chapter 2, right? And we said that 2 of the modified glucose molecules we introduced was N-acetylglucosamine, okay, here, and then N-acetyluramic acid here. So we're exploring the structure of peptidoglycan. So you guys, peptidoglycan is a polymer of alternating nag, NAG, and you guys, remember in cellulose how we had those beta-glycosidic bonds that made the long, straight cellulose fibers so that the cellulose fibers or filaments could pack tightly together and make lots and lots of hydrogen bonds, right? And that's what helps make them strong. Well, you guys, bacteria take, strengthening peptidoglycan to the next higher level. They're not going to rely just on hydrogen bonds. The bacteria are going to covalently cross-link the peptidoglycan. They're going to form covalent bridges or bonds between the peptidoglycan polymers. And the way they do this, you guys, I should have some covalent bonds here. They add a little, what we call a tetrapeptide tail to the end of this chain on N-acetyluramic acid. So tetrapeptide meaning there's 4 amino acids present. Okay. So let me just move forward here. And folks, if we look at page 3, this is a nicer cartoon. So you can see NAG and NAM, and attached to NAM is that little tetrapeptide tail. And you guys check it out. Well, L-alanine, okay, that's an amino acid we find in proteins. But you guys, what's the second one? It's one of those weird D optical isomers. Remember we said that in proteins we only find the L isomers? Well, here in peptidoglycan we find one of those wild D isomers of amino acids. And then the third one down, you guys, diaminopimelic acid. It's an amino acid, but it's never used in protein synthesis, right? So there's some wild amino acids here. And then there's another of those D optical isomers. So you guys, when I talk about the tetrapeptide tail, that's the little chain of 4 amino acids I'm talking about, right? The reason it's important is that the bacteria are going to use those tails to cross-link the peptidoglycan to make it so incredibly strong. Oh, no way. I said 1150. I feel so bad because those guys are like, we have to get this done. Okay, let's see what we can do, you guys. I know this is going to drive you nuts. Okay, so you guys, this is the polymers of NAG and NAM. Here's the tetrapeptide tails. Here are those cool covalent crosslinks here. These crosslinks, you guys, are absolutely essential for the strength of the peptidoglycan. If we can stop those crosslinks from being formed, we're going to kill that bacteria through osmotic lysis. So you guys, what's the name of the bacterial enzyme that forms these crosslinks? We're going to call it bacterial transpeptidase. Okay. So you guys, what's the name of the bacterial enzyme that forms the crosslinks? Transpeptidase. You will also see it referred to, you guys, as penicillin binding proteins. We're going to use transpeptidase. So the function of the transpeptidase is to form those peptide crosslinks in the peptidoglycan. Those crosslinks, you guys, are essential for strength. If we prevent the crosslinks from forming, the peptidoglycan weakens, the cell wall weakens, and what's going to happen? Osmotic lysis and death. So you guys, how will we stop those crosslinks from forming? We've discovered antibiotics that will stop those crosslinks from forming. And the most famous one, you guys, is the first, oops, sorry guys, I'm sorry. Oh, the reason I put this here, folks, is on the lecture exam, you see a cartoon similar to this, but I'm gonna white out the labels. I'll put letter labels on these ring structures here. So you guys, if I ask which of the letter labeled structures is N-acetyluramic acid, the key is, you guys, look where the little tetrapeptide tail is. It's always linked to name, right? And if I ask you where's NAG, you know it's the ring structure with no tetrapeptide tail. Okay. Remember, go ahead, sorry, you guys.
NAM is always on the tetrapeptide tail.
Is always on the NAM, yeah. And if I ask you what kinds of bonds links these two sugars, you're going to tell me a beta-glycosidic bond. If I ask you guys what kind of bond do we find here and here, what kind of bond is that? A peptide bond, right, between amino acids. Okay, and you guys, this is important. Why do I have stars on this peptide bond between the tetrapeptide tail of this strand and the tetrapeptide tail of this strand? That's the bond made by transpeptidase, right? So if we would stop transpeptidase from forming that bond, we can kill the bacteria. Yes? What was the first antibiotic discovered, you guys, by Alexander Fleming? Penicillin. Guess what penicillin does? It stops the peptide phosphates from forming because it binds to bacterial transpeptidase and shuts it down. Penicillin binds and that transpeptidase can no longer function. Right? So you guys, does this make sense? How does penicillin kill our bacteria living in hypoosmotic environments? It doesn't kill the bond. It prevents the bond from forming. Prevents the bond from forming. Does that make sense, you guys? Okay. And you guys, I'm not going to go through the history of the discovery here just because I don't know noise going on. But remember, Sir Alexander Fleming was the first to discover penicillin, the first antibiotic discovered. Penicillin, you guys, is made by which type of microbe? Fungus, yeah. And what's the name of the fungus? Penicillium, right? So penicillium makes the antibiotic penicillin that kills bacteria. So sometimes they're dramatic and say it's like chemical warfare amongst microbes. A bunch of trying to wipe out the bacteria, right? I don't know if that's true or not, but it's exciting. So you guys, these are some examples of beta-lactam antibiotics. We have penicillin, is like the grandparent, and then we have amoxicillin, ampicillin, methicillin. The cephalosporins have the same mechanism. So all of these beta-lactams, you guys, how will they kill the bacteria? Prevent those peptide crosslinks and glycan, and the bacterium dies from osmotic lysis. Awesome. Hey guys, I don't want you to worry about this right now. Vancomycin also prevents those crosslinks, but it's through a different mechanism. So just for right now, you guys, just maybe on the back burner, just remember vancomycin also prevents those peptide crosslinks from forming. Okay. Now another discovery, you guys, Sir Alexander Fleming. This was an amazing person. He discovered this enzyme that's found in the white part of eggs and tears and sweat. And this enzyme, you guys, is called lysozyme, right? Let me back up. I'm sorry, guys. I'm just going to back up here. See on your cartoon, see this beta-glycosidic bond? This beta-glycosidic bond is cut by lysozyme. It's part of our natural defenses to kill invading bacteria. So lysozyme, you guys, it cuts, it hydrolyzes this glycosidic bond between NAM and A. What will be the result? Will the peptide glycan weaken? It will. And how will the bacteria die? Osmotic lysis, right? It's just another way to kill the bacteria. This time we're going to attack that sugar backbone. Okay. All right. And that's lysozyme. Okay, folks, and you're going to kill me for this, but I just so want to squeeze this in so your brain's working on it over the weekend. So you guys, next week we're going to be doing gram stain. So we want to understand the history of gram staining. Okay, so you guys, gram staining was developed by a Danish pathologist by the name of Hans Christian Gram. And as a pathologist, he was trying to figure out, like, when people would die in the hospital, for example, his job was to try to figure out why did they die. Now, Gram knew about the germ theory of disease, that microbes can invade, hosts and cause death. So he was always interested to determine, did the patient die from a bacterial infection? But the problem is, you guys, bacteria, most bacteria in nature are not, they don't have pigment, right? They're clear. And they're so tiny, if you're looking at fluids of a patient or blood or tissue of a patient, it's so hard to see those colorless bacteria, right? So Gram was trying to develop a differential stain. What he was hoping is that he could come up with a staining protocol that maybe would stain the bacteria and not stain the surrounding human cells, so the bacteria would just be really obvious, right? So in the Gram stain, you guys, we started out with crystal violet, and a mordant called Gram's iodine. And if we look at bacteria, you guys, I'm gonna use two different types of bacteria, positive and negative. And in addition, you guys, we're gonna look at human cells. So as you will see next week, when you add your crystal violet and Gram's iodine to your bacteria and to your cheek cells, right? Everything is stained purple. I don't have purple, you guys, but everything is stained purple. And what Gram was hoping was that then he could add a decolorizer, a lipid solvent. And our decolorizer is alcohol acetone. This is called the decolorizer. And what he was hoping was the decolorizer would break down the cell membrane and all the stain would wash out. What he was hoping was that the cell wall of the bacteria would hold the color inside. So what he was hoping was the bacteria would be colored and the human cells would not. But what caught him by surprise today is that he discovered that some bacteria also decolorized. So initially he called these positive bacteria, Gram-positive bacteria. He called these negative bacteria, Gram-negative bacteria. But now we can't see those. So in our modern Gram stain, you guys, we add a secondary stain, a counter stain called saffron. So at the end of the Gram stain, you guys, Gram-positive bacteria remain protocol. Gram-negative bacteria will be what color, you guys? Pink or red, right? And what color will your cells be? Pink or red, right? Okay, and we'll leave you here, you guys. The reason we see this difference is that Gram-positive bacteria cells, have thick layers of active ligand that trap the violet iodine inside. Gram-negative bacteria, guys, they only have this thin layer of active ligand, can't trap the crystal violet inside. But next week, you guys, we're going to develop Gram-negative bacteria, though, have this really cool hydrophobic outer membrane that can protect them from a lot of our antibiotics. Okay, you guys, so I'm sorry that was so nice. You guys are troopers. We'll start here on the 3 different